Good afternoon, everyone. So I've always been told to start a talk with a joke. Um, at the sixth International Jelly Blooms Symposium, my first ever conference, I tried to start my talk with a joke, and it was met with deafening silence and a disappointed look from my supervisor. So there won't be any jokes this year. So I'm going to talk about morphoplasticity and environment, the lines between, where the lines between taxonomy and ecology blur. So while most of us will inherently understand the term morphoplasticity, Google apparently doesn't. If you try Google morphoplasticity, you'll get a bunch of different answers, so let's just break it down. So coined from the term morphology, which originates from the Greek morphe, referring to general aspects of, or in biological terms, it refers to the general aspects of biological form and the arrangement of parts of a plant or animal. Plasticity in a biological context refers to the adaptability of an organism to changes in its environment. So morphoplasticity is talking about the changes in an organism's form based on its environment. Why do we care? I was very happy when Alan put up a section of the ICZN code earlier in his talk. This is a section relating to holotypes and the fixation of names based on a name-bearing type. Ignore the part on the right-hand side. We'll get to that in a bit. So the idea of a holotype is very interesting in that in theory, it's what we go back to when we don't know. It's if we find something and we don't know what to compare, or if we've got data that disagrees, we go back to the holotype and we compare to that. And the idea of morphological description, so when I first started, was obsessed with genetic work. I was obsessed with computational biology, and I still am. But Andre actually made a comment on one of the first manuscripts I submitted, saying, well, what's the usefulness of the species description if you're not going to provide us with morphological characteristics that people can actually use in field? So I went back, did some work, came up with the species description there that actually worked. And it agrees very much with the philosophy that I have about if we're going to do science and we're going to put all this time in and get this funding, the science needs to be useful. And so it needs to be able to be applied by a wider range of people than just us in a lab. The comment on the right-hand side is actually, it came from my PhD review this year from one of the examiners, and I was slightly frustrated by it at first. I was actually a little offended because it seems to break down what we do quite simplistically, and I had a moment of, how dare they simplify our work so easily? And then you go and look at what we do fairly frequently statistically. So just to give you an idea, if we talk about holotypes and why we have problems in this area, thanks Mike for letting me use this next example. But if you were a young scientist and you arrived in Palau and you were looking at Mastigius, those two things for most people don't look the same. If one of those was your holotype, you may have a serious problem trying to describe the second as the same thing. So, and then on the converse, we have, well, not on the converse, actually agreeing. If you look at Aurelia, so we can distinguish Aurelia morphologically a lot of the time, hopefully most of the time. And if you want to see more of that, a great paper is Scarano's 2017 paper, where she describes Relicta. She has some very clear photos and descriptions of the differences between these. But what's very interesting is if you have a look over here at the top right-hand side of the CAP analyses, those blue triangles and the green squares, those are the same species. Those are both Aurelia from different populations, and they exhibit the same, almost more morphological variability sometimes than some of the other species that we examined at the same time. Not to say they couldn't be distinguished. They still fall, both groupings fall outside of the other species in the area or in the graph, but they display a huge amount of variability. So if we look at species delimitations, We know at the moment we have nearly 70 species concepts. That's not particularly helpful or useful for us a lot of the time. There's no unified species definition, and Alan and I were talking about this the other day. Part of the reason is that some species hypotheses and species concepts may be in complete opposition to others. And then I was reading some discussions on morphospecies. I went back to some of Mayer's work, and morphospecies were never meant to be, it was never meant to be a species concept itself. It's representative, it's diagnostic of a species. It's not meant to be a species concept in its own. And that comes back to what a lot of us are pushing about integrated taxonomy and integrating multiple lines of evidence. So when we talk about species, we often look at what we have with us at the time, and that can become quite confusing when you're doing species descriptions because species diverge over time. We know that this isn't a set in stone result that we're seeing at the moment. Things grow. This is probably one of my favorite pictures in science, the tree of life, looking at divergences of organisms. This is Darwin's original draw of the tree. And so we know these things happen. You look at, in the jellyfish context, we've got huge divergences in Aurelia over large geographical positions, and they are quite distinct at local level. We know that our environments affect species, so we've got Darwin's drawings of finches. I'm on a bit of a Darwin trip at the moment. But we know that these adapt and evolve for ecological niches. I don't know if Darwin would be particularly happy with me using his discussions in the concepts of species. He seems quite judgmental of the species concept. And so then we look at, all right, case of the West African cichlid. So in the space of time that humans and chimps diverged from our most recent common ancestor, there have been more than 700 divergences in West African cichlids. These fall within 3 major lakes in Africa. And what's quite interesting is genetically we see interbreeding between a bunch of these populations. So we're getting niche occupation. We are getting specialized species evolving, but we have seen fairly recent interbreeding between populations. And then we look at work that one of my colleagues did, a very brilliant colleague of mine. We look closely to home for me and Chrysora. So we have 3 or potentially 4 species along the west coast of Africa, but I find these very interesting because the ranges for Chrysora fulgida at the top and Chrysora galensis at the bottom are actually very, very close. In fact, at the extreme ends of their ranges, they overlap. Yes, we know that we get huge variation in color morphs and structure within these species. But if you look at those 2 species, they are fairly distinguishable. Now, the map probably belies how close these populations are, but I can, within the space of half an hour, dive in the home range of Igalensis and dive within the home range of Fulgida. In fact, I cover those ranges on my drive to the lab quite in the mornings. So then you ask, well, how diverse are those environments? So anecdotally, if I dive in probably closer to the home range of Igalensis here in False Bay, I can dive in water of about 18 degrees. And if I cross over the Cape Peninsula to the start of the Benguela upwelling region, which is where we find Fulgida, on the same day, I can have 8-degree water. So Agalensis is found in an area that we sometimes term the Goldilocks zone. So we have, as Delphine pointed out, we have the Agulhas current running down the coast here. It's a warm water current. We have the Benguela upwelling system, which runs up the west coast of Africa here, which is a very cold but nutrient-dense current. And what we have happening here in False Bay is eddies of warm water splitting off that then mix with this nutrient-dense water, and we get what's called the Goldilocks zone. But that's a slight digression. These species sit in fairly different ecological areas. which may be, and I've discussed this with Verena, may be part of the reason that we are seeing a divergence within the species. So with all this being said, where do we go from here? And this is probably the part of the talk that's going to make some people uncomfortable. My argument is that, that depending on what you want to do, that answer differs. So one of my favorite lines from a taxonomic paper comes from a paper that Mike and Lisa wrote a while ago, and it basically said that taxonomy is an expression of the human need to classify things. And so when we talk about species concepts, we really need to look at the use of those species. So if you're a conservationist and all you're looking to do is conserve an organism in your area, the morphology, if it's being defined by the area, is probably good enough to go on when you're identifying a species. If you want to look at whether an organism has invaded an area as an invasive species, you need to delve a fair amount deeper. If you're an ecologist, you need to make sure that what you are identifying is what you say it is, and so you need to put a lot more effort into your species descriptions. If we go one step further, we look at taxonomy, which is where I based my original philosophies on. And there we have to be certain. Mike spoke about making the boxes and then making sure we fill them with the correct things. We need to do this properly, and that's where the strengths and the development of integrated taxonomy is very useful. In that case, I'm looking at developing a machine learning model that will distinguish these species without the introduction of human on a morphological pattern, on a morphological level, so that we can then run genetics on a priori groups without having to make those decisions as humans. Our decision making is often fallible. If you're an evolutionary biologist, I think you should be telling me how to do this. It's your job to discern these sort of things, and we should be borrowing information from other fields like evolutionary biology when we make these decisions. And with that, thank you for listening to my talk.